Welcome to the Wright Science Daily Revision Boost. First thing, big congratulations to our top 3 scorers from our Day 9 quiz, which were Dewey, Aoife and Madeline. Well done folks, let's see if you can do it again this week. Today's Daily Booster is a separate science special. So for those of you doing combined science, then this is work you don't need to know. So stop the video, go and have a night off and enjoy yourselves. Those of you doing separate sciences, this is the stuff that you need to know that those doing combined science don't. In today's Biology Blast, we're going to have a look at the eye and the brain. So the first thing we need to know are the different parts of the eye. So hopefully you've sat down with the diagram and gone through to make sure you know them. We also need to know what each part does. So the cornea refracts the light to focus it. Then as the light passes through the pupil, we further refract it as it goes through the lens. And this creates a sharp image on the retina. And the retina itself has these photoreceptors which will produce a nervous impulse which travels down the optic nerve to the brain. What we need to be able to do is obviously focus on objects that are different distances from us. And in order to do that, our lens has to change shape. And this whole process is carried out by the ciliary muscle. So if we're focusing on a near object, the ciliary muscle contracts and the lens becomes more convex or fatter. And if we want to focus on an object that's further away, the ciliary muscle will relax and make the lens more convex or thinner. There are 3 key problems associated with our vision we need to know about. Short-sightedness, which is where distant objects appear blurry, and this is caused by the lens becoming too strong or the eyeball too long, and we correct it with a concave lens. Long-sightedness is where near objects appear blurry, and that's caused by the lens becoming too weak or the eyeball too short, and we correct that with a convex lens. Third one is colour blindness, and that's where people have difficulty making out the different colours, and the most common form of this is red-green colour blindness. Important to remember that colour blindness is a genetically inherited condition, and it usually affects males. So when we mentioned the retina has these photoreceptor cells, there are actually 2 types. There are rod cells, which allow you to see in low light levels, but they're not responsive to different colours, and the cone cells, which do respond to the different colours. And we've got different cone cells that respond to red light, blue light, and green light. The next topic we're going to look at is the brain. And the brain is vitally important because it's going to process all of the information collected by those receptor cells and from the hormonal system about any internal and external environmental changes and then produce a coordinated response to them. Having the brain as that central control centre makes the process much faster than if we had different functions controlled by different regions in the body. You do need to be able to recognise and label the different parts of the brain, so make sure you've tested yourself with that diagram. We have 5 main areas of the brain that you need to know. The cerebrum controls complex behaviour like learning, memory, etc. The cerebellum controls your posture, balance and any involuntary movements. The medulla controls automatic actions like your heart rate and breathing rate. The hypothalamus, which regulates temperature and water balance, and the pituitary gland, which stores and releases hormones that regulate a lot of body functions. For those of you doing the higher tier, you need to know about how we study the brain. So we've got a few different ways we can do this. We can use brain mapping from stroke patients. We can place electrodes into the brains of animals and humans. We can use 3 different types of scan, the CT, the MRI and the fMRI or functional MRI. We do have some difficulties, however, studying the brain that we need to be aware of. Firstly, patients have to give consent for their information to be shared, and some may not want to do this, as with certain conditions you may be deemed as somewhat of a curiosity. We need to actually analyse many case studies to be able to draw reliable conclusions. Each study that we do is an individual and therefore will have different results, making it quite hard to draw those conclusions. When we're talking about a specific function, then several areas of the brain may be involved in that one function. And many people also think that any animal testing that we're doing to ascertain this information is unethical as well. When we think about our nervous system, it's divided up into 2 parts. the central nervous system, or CNS, which is your brain and spinal cord, and the peripheral nervous system, or PNS, which is all of the neurons that connect the central nervous system to the rest of the body. Now, what we find is we can have damage caused to our nervous system, and typical causes of this would be injury, perhaps a genetic condition like Huntington's disease. There could be other diseases that lead to damage. We could have toxic substances being consumed. All of them will have some kind of damage on our nervous system. If it's damage to the peripheral nervous system, then that's going to be affecting the sensory and motor neurons. So this might result in your inability to detect pain. It might lead to numbness or a loss of coordination. If we're talking about damage to the central nervous system, then we could see the loss of control of entire body systems. You might have partial or complete paralysis, and there may also be memory loss or processing difficulties depending on where has been damaged. There's a big problem with actually trying to repair the central nervous system damage. First, if we think about the spinal cord, then identifying the individual nerve fibre that's damaged and repairing it without damaging all of those others around it is very difficult. So normally spinal injuries are irreversible. If we think about brain damage, number one, it's very difficult to diagnose, and then we've only got a few treatments that we can actually resort to. So radiotherapy or chemotherapy for cancers, surgery, and deep brain stimulation. In today's chemistry check, we're going to have a look at the transition metals and how we can identify different chemicals. So the transition metals are the ones that are found between groups 2 and 3 on the periodic table. Transition metals are good catalysts, and they're metals that react very slowly, if at all. What we do find though is that they are capable of forming more than one type of ion. So what we have to identify what charge the ion has is a little Roman numeral in brackets. So make sure you do know 1, 2, 3 because they're the ones that are likely to come up. We also find that our transition metals produce coloured ionic compounds, whereas the alkali metals tend to produce white or colourless compounds. If we want to detect cations, we need to understand what happens to metals when they burn. So the first thing is that the metal ions are vaporised, and then they're going to absorb energy as this happens, and the electrons within their structure jump to higher electron shells. Then as those electrons drop back to their original electron shell, it's going to release light energy to the surroundings as radiation. In order to actually identify some of these cations, then we can carry out a flame test. So in order to do this, we get a nichrome wire loop, dip it in concentrated hydrochloric acid, and then hold it in a Bunsen burner flame until the colour doesn't change. You then dip it into your test powder or solution, and if it's a powder, make sure that the loop is damp first so it picks it up, and then hold it in the roaring flame and note the colour. There are 5 different metal ions we need to know the flame test colour for. Lithium is red, sodium yellow, potassium lilac, calcium is an orangey red, and copper greeny blue. Just be aware that in this experiment a main source of error is contamination. A second test we can do to detect cations is using sodium hydroxide solution. So when we add the sodium hydroxide, then a precipitate is formed, and a precipitate is an insoluble solid. The colour of that precipitate will tell us what metal ion we have. So iron 2 is green, iron 3 orange-brown, copper is blue, calcium white, and zinc is white. So the way we actually work out whether it's calcium or zinc is add an excess of sodium hydroxide, because calcium hydroxide will not dissolve, whereas zinc hydroxide will dissolve. The next 2 tests we're going to look at are ones to detect anions. So if we want to detect if sulfate ions are present, then we add a few drops of dilute hydrochloric acid, then a few drops of barium chloride solution, and if a white precipitate forms, then we have sulfate ions present. If we wanted to test for carbonate ions, we add a few drops of hydrochloric acid, and if bubbles of gas are made, then we've got carbonate ions present. The last type of anion we could be asked how we detect are the halide ions. So the way we do this is add a few drops of nitric acid just to remove any other ions present to avoid a confusing precipitate, and then we add a few drops of silver nitrate solution, and the colour of the precipitate tells us which halide ion is present. So if it's white, it's a chloride, if it's cream, it's a bromide, if it's yellow, it's iodide. In today's physics prep, we're going to have a look at pressure. So the first thing we need to know is the relationship between pressure and volume, and they are inversely proportional. So this means that if the volume doubles, the pressure will halve. And the reason for that is that the collision of the gas particles produces a force at right angles to the surface they collide with. So that if we halve the volume, then we're going to have twice the number of collisions each second with the container, so the pressure is going to double. If we think about how we can change the internal energy of a gas, then we can increase that internal energy in one of two ways. We can either heat it or we can do work on it. The second type of pressure we need to know about is atmospheric pressure. So the gases that make up our atmosphere exert this atmospheric pressure, and on the surface of the Earth it's about 100,000 pascals. As we move away from the surface of the Earth, then atmospheric pressure decreases, and the reason for this is there are fewer particles of air pushing down on us. One thing to bear in mind though is when we're using a pressure gauge, it will always read atmospheric pressure when it's turned on, not 0, so you have to set it to 0. The third type of pressure we're going to look at is for the higher tier only, and it's liquid pressure. So water exerts a liquid pressure, and this liquid pressure will act in all directions. What we find is as we go deeper down in the liquid, then the pressure will increase. And the reason for that is we've got more particles above us pushing down. Second pattern to bear in mind is that the denser the liquid, the greater the pressure exerted at the same depth will be. So that's due to there being a greater weight of liquid pushing down in the denser one. Now we've had a quick recap, then head on over to socrative.com and test your knowledge on the topics from today. The room name you need to enter is Wright 2662. Good luck! As always, if there have been any topics that we've gone through quickly tonight that you're not 100% certain on, then head on over to the playlists for more in-depth discussions on each topic. So for biology, it's the B3 playlist. For the chemistry, it's the C4 playlist. And for the physics, it's the P1 playlist. And remember to make sure you're looking at the biology, chemistry or physics playlists, not the combined.